A very good morning, students. So, as you know that we are revising our chapter Fundamental Unit of Life, and in my last lecture I promised you that we will draw the structures, right? Because many times I feel that many students they don't like biology because they feel that they cannot they do not have a good drawing, they cannot draw structures, they cannot explain few concept, but I am trying to make it very simpler for you, right? So in this special lecture, in the I am trying to make every diagram from this chapter, make it simpler for you so that you can easily draw them in the your exam, right? So here the question is, draw a well, this question sometimes it is asked in 3 marks, that draw a well labeled diagram of a eukaryotic. You have to pay concentration that you are asked about the nucleoid, because nucleoid is present in the bacteria, right? And you are asked about the nucleoid, or you are asked about the eukaryotic nucleus, because particular nucleus, well defined nucleus, is only found in the eukaryotic cell. So how to draw it? Let's draw first one circle, right? Just make you have to draw uh, one outline so that you can easily draw the outer membrane. Now always remember that the membrane, or I will say the outer membrane, outer nuclear membrane in the nucleus, in the eukaryotic nucleus, it is not continuous, right? So you can draw it like this. This is, in this way you can complete this whole circle. You have to leave few gaps in between, and in this way the outer membrane is present in the eukaryotic nucleus. So we are going to make it like this only, and you have to complete it like this. It is present with the gaps. So this, let's label it first. This is our outer nuclear membrane. Clear? Now the major portion which is present here is in the corner, in not in the center, in the corner on the side there is present this round nucleolus. This nucleolus is present here, and then there is these thread like structures you can observe in the nucleus, thread like network, and this thread like network is known as, till the time they are not condensed, these thread like fibers, they are known as chromatin fibers. Now definitely, because we are drawing the nucleus, so there will, in the cell like, there is a jelly like material. So in the nucleus there is a substance which is present, that is this substance which I am right now drawing, that is called your nucleoplasm. Just like in the cell cytoplasm is present, here you have to mark nucleoplasm, right? Now you will ask, ma'am, what is these gaps which are present here? These gaps are your, these after nuclear, these gaps, they are known as nuclear pores, right? So chromatin fibers, outer nuclear membrane, and this is the outer nuclear membrane, and this one is inner nuclear membrane. The outer part is known as outer nuclear membrane, and this portion is outer, and inner portion which is towards the inner side, that is called inner nuclear membrane. So this is the structure of your eukaryotic nucleus. Now the next part, so first part is cleared. We have drawn the label structure. How is it different from nucleoid? Now See, nucleus is different from nucleoid in many ways. We will mention, because it is a, if it comes in 5 marks question, we are going to write each point. So how is it different from nucleoid? You know that nucleoid is present in the bacteria. First thing, it has a membrane. So we will write that nucleus is a membrane bound organelle, right? It is a having a membrane, so we will write that it is a membrane bound organelle. But membrane covering, or you can say membrane, is absent in nucleoid. I think first point is very much clear to every one of you, that membrane is present in nucleus but absent in the nucleoid. Nucleolus, we have drawn here nucleolus. It is present in nucleus but absent, it is absent in nucleoid, right? The third point, the size. If you look at the size of nucleus is larger than न्यूक्लियॉइड. इस न्यूक्लियॉइड से थोड़ा सा कंपैरेटिवली बड़ा रहता है न्यूक्लियस का जो साइज. नाउ डीएनए कंटेंट ऑफ न्यूक्लियस इज, जो सिंगल डीएनए कंटेंट है न्यूक्लियस में, सिंगल न्यूक्लियस में, इज इक्वल टू मैनी डीएनए मॉलिक्यूल्स. मैनी डीएनए मॉलिक्यूल्स, इतना ज़्यादा है डीएनए यहाँ पे, मैनी डीएनए मॉलिक्यूल्स एज कंपेयर टू सिंगल. in nucleoid there is only single DNA as compared to single DNA in nucleoid. Means that जो DNA, these chromatin fibers, they are equal to many DNA molecules. But जो nucleoid में we are observed only and only single DNA. So these are the 4 points you can very confidently, first thing you can draw this diagram very easily and you can write down these points. So your you can say that another question with the diagram, it is ready, right? It is ready for you. Now comes to the second question, because we have talked about now nucleoid. So now let's talk about the structure of the prokaryotic cells, right? Because we have drawn now we have talked about this nucleoid structure. So let's next question, draw a neat and well labeled diagram of prokaryotic cell, right? If this question comes in exam, then how you are going to draw the diagram? Let's draw it very easily. First, let's make the outer covering. We are going to make it little oval shaped kind of structure. So here you can see we can draw this outer covering first. Don't go on my irregular pattern, you can draw it clearly, right? So now this covering, that is known as what does prokaryotes they have? Prokaryote, they have which structure? This is known as cell wall, right? This is, we have drawn this outermost covering, we have drawn here is, that is the, let's label it with the another, this is cell wall, right? This is the cell wall. Now after the cell wall, what will come? There will come this plasma membrane. But right now with the plasma membrane, I am making few another kind of molecules also. right? So this plasma membrane, this is the, label it as the plasma membrane. In at few points, plasma membrane is modified, right? As few points, plasma membrane is modified. It is not directly, or I will say continuously present. It is modified at few points, and these modifications are known as mesosome, right? This is known as mesosome. Then inside this, there is, you will found ribosomes. So let's label it. These are ribosomes. Now there is the another thing which is found here, that is this kind of structure. All through the, few are small, few are long, few are small. These kind of projections, they are known as, which are found in our bacteria cell, these are called fimbriae. These are called fimbriae, right? These are called fimbriae. And then they also have, there is the present these long extensions, and these are called flagella. This is known as flagella. And inner side of this, at, you can say, this structure you find at the corner, or the in the middle actually, that is called nucleoid. This is known as nucleoid. And then it is completely filled with the cytoplasm. Make it the cytoplasm. So this is the well labeled structure of the prokaryotic cell, or you can say bacteria, which you can draw very easily in your exam. Is it difficult? Do you find that it is difficult? Now one more major thing which is not given in many books, which you should know actually. This bacteria, it is covered on the outer side with a covering, with very thick, I am making right now, with very thick, this is very thick and tough covering of the bacteria. Every bacterial cell is covered with this, that is the, this thick and tough covering. Even beyond, after the cell wall, it is present, and this thick and tough covering is known as capsule. It is the speciality of the bacteria. So let's make it with, I'm labeling it, this is known as capsule, right? So first thing, first capsule, then cell wall, then plasma membrane. There is the modification of plasma membrane, which are known as mesosome. Then there is present, these are the ribosomes. Then there is the cytoplasm. Then there are the fimbriae, and there is the, for locomotion, there is present flagella, and in the center we are having nucleoid, which is not covered with any kind of membrane. I think you can easily draw now this structure of the prokaryotic cell as well as the eukaryotic cell. Third question which I am taking now with you, that is the draw. ये structures जो हैं, these are very simpler one, and you can easily, very easily you can draw them in your you can say in during the exam. So draw a well labeled diagram of plant cell and label it its parts. Because many times I have observed that the diagrams which are given in your book, sometimes just because you are not able to understand them, so you don't want to draw them, right? So here let's draw the plant cell in a very easy manner. First thing which is important for a plant cell to draw, just make its outline, right? So here as you can see that we are making outlining structure. Let's make it like this. Plant cell we always make like this, right? So you can take the help of your ruler, because I am drawing it as a free-handed right now, but you can draw it in a very beautiful manner, right? So here you can see that we are first drawing the outer lining of our plant cell, right? Now let's make the inner lining. Let's make the inner one. Okay, now cover it with the second covering, that is, and this is what we are drawing, that is the known as your cell wall. Do you find cell wall in animal cell also? Will you be able to find cell wall in animal? No, there will be, I'm really sorry, it's okay, there will be no cell wall in the animal cell. So let's make it clear so that we can have distinguish cell wall of our plant cell. Make it all with the same width, that will be more specific, that all sides they are from the same width, एक ही चौड़ाई जैसे बनाना है, but because I am drawing here free handedly, so you can draw it with the ruler. Okay, so this is, we are going to label it. You can also draw with me in your notebook one by one, right? So there we are done. That is, let's label it. This is our cell wall. Now we are moving ahead. After cell wall, what will you observe? After cell wall, what will you find? That is your plasma membrane. So let's draw plasma membrane. So inner side, once you will start moving, inner side you will find this plasma membrane, right? Once you will move inner side, first सबसे पहला काम क्या करना है आपको, ड्रॉ करनी है cell wall, and after that you have to draw plasma membrane. So here we are done with the plasma membrane, right? Now the major, another very beautiful thing which is present, which actually occupies the major space of plant cell. कौन ऑक्युपाई करता है मेजर स्पेस प्लांट सेल में? दैट इज वैक्यूल, राइट? दिस इज कॉल्ड वैक्यूल. This is vacuole. Now many students, many times they are very confused that what is present inside this vacuole. Actually inside this vacuole we are having sap. Now what is sap? Definitely that question will come in your mind ki ma'am what is sap? Actually sap is what? It includes water plus minerals. So our many things are done. We have drawn the cell wall, we have drawn the plasma membrane, we are done with our vacuole. Now there is another very important structure which is not present in animal cell, which is very helpful for the plant cell to make their own food. Can you guess it? Which helpful for the plants to make their own food? Can you guess it? This is what I am drawing right now. This is chloroplast, because chloroplast is having chlorophyll and it is helpful in the photosynthesis, right? So chloroplast is done. Now After the detailed structure of chloroplast, definitely we will draw later. Now let's draw the nucleus. Again, you have to be careful that nucleus, when you're drawing nucleus, we will draw it like this only, with the double membrane structure, right? We are going to draw with the double membrane structure. And here in the nucleus, what will you found? This is the nucleolus, these are the chromatin fibers, and then this is the nucleoplasm, right? So label it. Let's label this. This is our nucleus. Just very closer to nucleus, there arises this network. ये बुक में क्लियरली नहीं दिया होता है तो बच्चे इसको ड्रॉ नहीं करते, राइट? दिस काइंड ऑफ नेटवर्क यू हैव टू ड्रॉ, एंड दिस नेटवर्क इज कॉल्ड your endoplasmic reticulum. And now we know that one I will drawing here, right? One I am going to draw here so that you can clearly understand them. Now just look here, that on this let's make it with another pen. So here on this endoplasmic reticulum you will find these small tiny dots. And what are these dots? These are actually ribosomes. And the endoplasmic reticulum with ribosome is known as rough endoplasmic reticulum. And now there is no ribosome. So this is known as SER, smooth एंडोप्लाज्मिक रेटिकुलम. और हमेशा जब भी ये बनाने हैं ये न्यूक्लियर मेम्ब्रेन से अराइज होते हैं. ऑलवेज रिमेंबर दैट इज़ अ थिंग दैट यू हैव टू रिमेंबर, दैट जब भी एंडोप्लाज्मिक रेटिकुलम बनाना है उसको हमेशा वैसा ही बनाना है, क्लोज टू द न्यूक्लियर मेम्ब्रेन. नाउ हियर लेट्स ड्रॉ दिस इनफोल्डेड स्ट्रक्चर. लास्ट टाइम हमने पूरा डिटेल स्ट्रक्चर ड्रॉ किया था, दैट इनफोल्ड्स which are present in the matrix. So this is our mitochondria. That is what we have drawn here, mitochondria. One more organelle which is found here in the plants, that is the, let's make it with the another color, that is these kinds of bodies you are find with the small circular, these kind of pattern, right? This pattern, one big, then there is this kind of face. Always make it like this. This is known as your Golgi body. This is known as our Golgi body, right? And there is a now gap. This actually this is not continuous. This is like there is a gap. Then this goes like this. Same, this membrane, that is your cell wall, it has a few gaps, right? And these gaps, they are known as pits. So you can make it whole like, so there are gaps in between these. So these gaps, they are known as pits. Pits hota hai pores jiske through material can exchange. Now here is present another structure, round structure, bodies. These are our lysosomes, right? These are lysosomes. And then there is present our jelly-like structure. And what is that structure is known as? This jelly-like substance, it is known as your cytoplasm. So that covers, that completes your plant cell. I am very confident that after watching this video, at least your fear that you cannot draw, that will be gone. That will definitely be gone. I don't think that this structure nobody can draw. You can easily draw these structures. So what we have drawn today, that is the eukaryotic nucleus. then there is the bacterial cell, and one is the plant cell, which is very easy to draw. In the next continuing with this video, I will draw animal cell as well as we will draw the Golgi bodies and the endoplasmic reticulum with you, right?